participants already know about NIME and what is their experience with NIME. It would be nice to hear. And please make this interactive. I'm sure there will be a lot of questions coming up, but I thought to understand you better before the session. So what is your understanding about NIME, and have you already installed NIME in your machines? The links were being provided. So are you able to install it? Hello, I'm speaking to you guys. Okay, you are able to install. I can see a response from one person. Okay. Anyway, so I think slowly you will get into interaction. Yeah, because unless you tell me things, it will be difficult. So let me share my screen. Software is complicated to use, so couldn't explore. Don't worry, we'll make it simple. Yeah, it looks complicated, very true, but don't worry. It's so simple as it is. Okay. So the first session, we will go with a few slides, and we will discuss in detail about those things. And then we will get on to the practical sessions. To be honest, in my GitHub, I have not shared any of the workflows, but I have shared the pictures of the workflows. And there's a reason behind it, which I will discuss at the end of the slides. Anyway, so the materials that is being presented in the slide might contain pictures or videos or codes or materials that is taken from public resources, which has a right to use, and also copyrights and ownership goes to the respective authors. I cannot create everything by myself. Of course, I have adapted from many other codes, to be honest. But many of the NIME workflows I have created myself. And multiple resources IDs are being used. So make your right choice. Today, we will be looking into NIME software, the platform, then Orange, as well as a combination of Anaconda and Python. And also, I don't declare any conflict of interest and does not have any connection with my employer-employee. And slides, whatever I'm sharing today, are shared via SlideShare. I will share the links at the end of the slides presentation. The codes, especially the NIME workflows and the Orange workflow, they are all shared in the GitHub. The links are already there in the shared schedule or titles. And it could be done in Colab also in certain cases. And yeah, I have generated MD simulations on GPU using Google Colab, even for GROMACS as well as for NAMD. So you can run molecular dynamics on your computer, on your mobile phone, if you have access to Google Colab. And some of the more detailed videos are also shared via YouTube. Just to start with some of the codes in AI or even in drug discovery, AI is clearly stimulating the drug discovery market and providing new opportunities for companies using such technologies. But the importance of ML technology is likely to increase in the coming years. So don't worry too much that I'm not knowing too much about ML algorithms. Am I staying behind the competing people there? The problem here is you're not actually competing, because you don't have much openings for ML in drug discovery. Still, it is on the research phase. Of course, there are many postdoctoral openings as well as PhD openings where they are asking for ML. But ultimately, ML is a very small portion of the study. The bigger portion is you have to generate data, right? The biggest drawback in drug discovery for ML is that we don't have sufficient curated data. We have a lot of data, but curated data is missing. If you tell me, sir, I have Campbell, okay, what do you do with Campbell? You have structure and you have activity data. What else you have? So many things are missing actually. So that's why when I discuss with data scientist guys, they used to tell me very difficult to work on drug discovery or other things because we don't have a ready-made data for us to use. something that we can get for like patient data or some disease data, population data, which is available in Google data sets or public data sets, even in Kaggle. We couldn't find much. There are very, very few. So we cannot do an extensive ML or a deep learning models using those data because we need tremendous amount of data. It is possible. We have to sit and extract from the literature and make curated data. So that is something that is yet to come, and it is likely to increase in the coming years. So get ready with whatever you want to learn. But keep in mind, as a biologist or as a chemist or as a drug discovery scientist, you need not too much worry about coding, right? If you really want, yes, you can be a developer. But if you want to be a developer and if you want to understand about coding, ensure you know about algebra, you know about statistics very thoroughly. ML is basically flooded with statistics. If you do not know statistics, there's no point in talking about ML in your any of your vicinity. So that's very important. So there are more than like very big companies, small sized, very small, mid sized, and large sized companies across the globe, which is all related to drug discovery. It includes CROs, it includes biotech companies, it includes software companies, or even AI companies. These are all getting involved, and there are many and plenty of them. So, Dr. Abraham mentioned, I'm a visiting scientist to Nairo Research India. So, Nairo Research India is also focusing on ML in drug discovery, as well as on aging and longevity. So, the main focus of the company is focusing on generating models for Indian population longevity. Of course, certain aging parameters and others being used. We have already published few papers on the molecular level. Now we already started generating models, and it's in the testing phase because we want to see how it likely can be reproduced and reliable. Now, this is being taken from a drug discovery report from the analysis that CAD in drug discovery likely to cost saving analysis to be done. That is where AI and ML is getting implemented. And cost saving analysis, we are able to save more cost as the years comes in and technology grows. And the upcoming trends in drug discovery is also like artificial intelligence, which includes deep learning, machine learning. For example, for deep learning, we are looking for data imputation. I'm sure many of you who are working on QSR or chem informatics would have come across that you will be having missing data of the activity data of your compounds. See, in order to, what do we usually do with missing data? We try to do some filling of data or trying to put 999 or minus 999 based on certain methodology or based on certain approach. But then coming to data imputation, it will try to train itself to try to predict those values and fill in those data for your better model predictability. So those techniques all came in. And it's also mainly said CAD is only being used in drug discovery process. However, the overall efficiency is likely to increase if CAD can be used for further developmental activities, and such as machine learning and AI. And there are a lot of CAD service providers. But if you see recently, for example, post 2015, you can see more than 25% it is being new CAD providers are getting established. And when you look at the CAD providers with approaches, most of them are on structure-based drug design. So it's mostly computational chemistry, but very few on fragment-based drug design and also on ligand-based drug design. But keep in mind, when you're doing fragment-based drug design, it's fancy, but make sure that you have an organic chemist or a medicinal chemist sitting next to you. If you're not an organic chemist, in order to design the fragments and to do the de novo synthesis, design. And when you're coming to structure-based design, keep in mind, just looking at docking score is not going to help you. Any compounds can dock with your molecule. If you have considered the whole chemical space available in this world, which is around 17 billion compounds. If you dock that and if you rank based upon the score or affinity, I agree to that. But you're not. You're trying to take a small space, maybe 10,000 compounds or a database of 4 lakh compounds. Within that chemical space, you're going to rank your docking score. So how can you say that that is the best molecule? Because you are not still explored the other chemical space. So it's very important that just don't give too much importance to score and affinity. It's more important has to be given on the biochemical factors. Understanding the mechanism, which is the amino acid, is very much important for your interactions. So based on that, you have to make decisions. When you're coming to ligand-based drug designing, again, there is a catch. You have to make sure what kind of data you have taken. If it is a pharmacophore analysis, are you taking a co-crystal ligand as your reference? Because the bioactive conformation, the 3D bioactive conformation is something that you have to be considered for your 3D QSCR descriptor calculation, as well as for your pharmacophore reference molecule. If you're not considering a bioactive conformation, that means if you don't have a protein structure in order to analyze that, then it is still a biased situation. Based on several confirmation analysis, you have to come up. But still, we have some error chances there. The standard deviation is always there in that prediction. And when you look at the CAD providers, service providers, you can see lead optimization is a hot topic there. That's where the brain comes in. Because lead optimization, human intelligence is to be put. You need to have a better understanding which functional group replacement is going to improve the activity or stabilize the toxicity or any other ADME properties. Then we have something called as hit to lead. So hit molecules are those which we can get from the potency data or even from the docking result. We just get the hit molecules. All these hit molecules can be transformed to lead if we find they have satisfying pharmacokinetic and pharmacodynamic properties. So that's where hit to lead comes into the picture. And many companies are still working on hit generation. That means just docking, virtual screening in the right way. And then many new companies recently came with the AI ML in target identification and validation. But for the general community or the scientific community, we have platforms like String, Stitch, DisGNet. They are all having target identification and validation steps. Of course, you can use Cytoscape for that. Now, we have to also understand what is the cause of the failure. In earlier days, just between 1960s and 1980s, the main reason for the failures of oral drugs was because of like pharmacokinetics, ADME, and efficacy. The other were not much great issue because human intelligence was put much into it because we didn't have any sophisticated SAR, that is structure activity models, relationship models, or even any softwares to play around with too much options. And later in between 2000 and 2010, most of the drugs that was being analyzed in the clinical trial failed just because of toxicity and strategy. When you're coming to strategy, let me tell within our scope, everybody use Lipinski rule of 5, right? I hope everybody knows about Lipinski rule of 5. Yes. If you know, how many of you have read the paper? If the answer is yes, you can type number 1. If the answer is no, you can type number 2 on your chat, in your chat actually, because if you cannot unmute and speak. How many of you have read the paper for Lipinski rule of 5? Okay, many of you are saying no. But hope you know what is Lipinski rule of 5, right? I will discuss about it, but I hope that you know it. So without knowing what is Lipinski rule of 5, just seeing what is a rule, why did you apply in your projects? Because you have not read the paper. So that is called wrong strategy. We need to apply something which we know thoroughly well. Lipinski rule of 5 is clearly said it is for oral bioavailability. And if you're so strict, I will take only compounds less than 500 molecular weight. Do you think that a molecule with 501 molecular weight is going to be bad? How many of you think the molecular weight that is with 501 is a bad molecule because Lipinski rule is not satisfied? If you think it is bad, number one, yes. If you think it is not bad, you can still consider, you can give number 2. So Lipinski rule of 5 says that molecular weight should be less than 500. So if I have a compound with 501 molecular weight, how many of you think it is bad and how many of you think it is good? Still can be considered. If it is bad, you can give number 1. Or if it is still good to be considered, you can give number 2. I could see here only 3 people have responded. So, or 3 or 4. Maybe others are a bit confused whether to say yes or no, right? So this is where the decision making is very important. So computational tools actually helps you to make decisions. They never tell you this is the best drug molecule. So please keep in that mind. And the computational tool, let it be any tool, let it be 100 million paid software or even a freely available tool, let it be any tool. Ultimately, we are trying to find out non-promising compounds from our data set. That is very important. And the confidence level. So many of you have said here, it is still good to consider the compounds with 501. You're absolutely right. There is no wrong in not considering, in considering the compound with 501 molecular weight, because we still have FDA approved drugs, which is having 750 molecular weight, even 852 molecular weight also. So what's wrong? So do not use unnecessarily Lipinski rule in unnecessary places. Because I have heard from many of the researchers that first task is that try to do some similarity search or download the database and try to filter them based upon Lipinski rule. Very wrong strategy. Don't do it at the initial stage. You can do it if we are very sure that your binding pocket of the protein has a limited space or shape. So only a specific molecular weight could fit in. And then you can do a trial and error method to see a specific threshold of molecular weight or ring number of rings or number of sugars that to be considered within your ligand in order to filter the database. If someone knows about coconut database, of course, we have some certain filtering criteria there. So you have to be very sure at what stage you filter with what filters. It's very important. Unnecessarily, if you do that, you're going to miss your key compounds. So when you're identifying hit, the data required for hit molecule is huge. That's where we call it as virtual screening. After finding out the hits, in order to get into lead, that's where we apply filters. So the initial stage, you have to use your filters very much cleverly. Otherwise, you're going to lose compounds. That's why we have to also have to look is machine learning overhyped. See, why I'm talking about machine learning here? Because I'm using machine learning approaches for QSR models that we are going to work today on 9 as well as on Orange. Even for Anaconda Jupyter Notebook also I do have. If time permits, I'll try to show that. So they took a survey in 2018. Things have changed now. Since last lockdown, I'm sure many developments have happened in machine learning. Even I myself have learned a lot and implemented a lot. I could see that many are citing my Jupyter Notebook, which was used for molecular dynamics using GROMACS, and many are making new versions of those notebook with much more advanced features. I'm so happy that everybody is making use of the lockdown, at least for upskilling themselves. So about For example, 13.9 percentage, including 31. So it's altogether almost 50%, I could say. Altogether, they are agreeing that machine learning is overhyped. And I also do agree that machine learning is overhyped in chemistry. It is not that easy for everything. Certain cases, it is really helpful, not in every cases. But still, we have some success stories like this. Exploring chemical space, can AI take us where no human has gone before? It is possible. But the question is how far these design molecules are synthesizable. That we have to wait and see. There are chemical spaces currently available from NME, from Wuxi, then from many other vendors, Molport and others. Those are about 80% synthesizable from the predictions. Now, when AI takes into it, we really don't know how far the synthetic feasibility or how far it is really synthesizable on bench. Another thing is that in silico medicine, very, very hyped, and of course, they're doing a lot of the analysis. So they came up with a molecule and a claim that within 46 days, they were able to identify a potential kinase inhibitor completely using AI. And it has entered into clinical trials. We have to wait and see how things goes. So we also wanted to see how the ML workflow is in chemistry. But before going to this slide, I also wanted to understand from different on all the participants, you're all from which domain? Chemistry or biotech or bioinfo or microbiology? Could you please tell me? That could be really helpful so that I can pitch accordingly. So what is your domain? Most of you, and most of you are students or faculties. Okay, mostly biotech, microbiology, zoology. Okay. So I shouldn't focus too much into chemistry then. Okay. Pharmacy is also there. Oh, hi, Abhijit. Okay. So bioinformatics, biochemistry. Okay. Okay. So that's good. I try to understand you guys. Okay. I will stay away from a little bit of too much into chemistry. But unfortunately, drug discovery is anyway chemistry. Yeah, it's more of bioinformatics, biotech, biochemistry, bioinfo, and few pharma. Okay, okay. I thank you so much. That helps me a lot. So before moving forward, let me also bifurcate it because we have a very diverse audience here. In the drug discovery, in the initial phase is called target identification, right? Where we have to identify the right protein or the gene target or the mechanism and identify the key amino acids, which is responsible for the upregulation or downregulation. So these all can be done by a biotechnologist or a bioinformatician. That is their main role, and where they can do network pharmacology or even systems biology analysis. So both the biochemist, biotech, or even bioinformaticians could help in that region. Later, it comes to lead discovery or lead optimization. Personally, my master's was on bioinformatics. So I think I would be the right person to claim to also correct this. Drug discovery is not completely bioinformatics. Sorry to say that, and that is the fact. Because when you come to drug discovery, you need to have a thorough understanding about chemistry because you're playing around with chemical compounds. But I already told you what is the role of bio people here because you are the main people to identify the target. Until unless you identify the target, there's no meaning of rest of the process, right? But then once it enters into the drug discovery or docking, most of the biologists will be able to screen a database, identify the best interactions, buy the compound, test it. Absolutely fine. But when it comes to lead optimization, our hands are tied. Because lead optimization means we have to make modifications in the compound. Now, if you think, sir, software does the modification, it's very easy and it is giving better activity. Now, the question is, can you test it in labs? Someone has to synthesize it, right? Who will synthesize it? Did someone confirm that whatever you designed on computers are synthesizable? No. If it is confirmed, then good. So that is something is lacking. So when a biologist works on these kinds of drug discovery project, ensure that you have a organic or a medicinal chemist sitting next to you, or pharmaceutical chemistry, because it's very important. And why I mentioned medicinal chemistry? Because if you're making a replacement of methyl to carboxylic acid, the person who is making changes should have an understanding whether this is going to affect the solubility, whether this is going to increase the permeability. All these things are very important. So those kinds of understanding is very important. Now you might think that we can achieve from literature. Many of the literature data cannot be transformed to wet lab when you're trying to come up with a new scaffold. It depends actually. The combinations or changes of the neighboring atom is also going to change the property. So that's what we have to keep in mind. That's where the pharmacologist will come into the picture. The pharmacologist will understand more about ADME, toxicity, not about the too much into mechanism. So ultimately, when you look at drug discovery, I have covered all the particular people who are in this session, including zoologists. Of course, you have to take care of toxicology, right? Toxicity studies and other animal studies has to be taken care of there. So everybody has a role. So if all join hands together, then only a drug discovery project can be complete. Now you might think, sir, is that possible? It is possible, but our mindset should change. That's the only thing. So make sure that when you're starting a drug discovery project, you have a good team who can discuss and who are experts in respective field. It's very, very important. Please keep that in mind. Everybody has a role, but keep position it properly so that you will get into the right decision. Because I have faced difficulties when I did my project in bioinformatics, because I was working on drug discovery that time, drug design basically. But yeah, and now I'm not interested to show my thesis of masters to anybody because I did a lot of mistakes there. So that realization is very important. Anyway, so considering all the fact, the multi-disciplinary audience is here. So I'll try to pitch in accordingly. Okay. So when you look at machine learning workflows in chemistry, of course, let's imagine that you identified some compounds that has to be synthesized, and it is being synthesized for the, and also looked at different screening or synthesis or different simulation data is available relatively for same compound. Of course, you need pre-processing. For example, data transformation, data wrangling, these all things are required because certain categorical data, computer machine learning algorithms cannot understand. So at that time, what we have to do, we have to transform it to numerical data. For example, you have a compound active and inactive. Computers never understand what is active and what is inactive. So to make it to understand, we say that active is 0, inactive is 1, or vice versa. Active is 1 or inactive is 0. Zeros and ones are understandable. So like that, these kinds of transformations and data wrangling is very important in machine learning. Similarly, which we used to do in QSAR, normalization, data normalization, we used to do that. And then we try to apply all the correct possible methods. Why I said correct? Because categorical data, classification data, numerical data, each one have selective algorithms or approaches that can be used from machine learning. You cannot use everything everywhere. So that also you need to know, aware about what kind of algorithm is possible for certain types of data. And then we can train themselves. But what is the main difference between statistics and machine learning? See, so far, whatever I told you is same with statistics also, right? Because we have multi-linear regression models there, then Bayesian model. We do have, again, partial least squares, right? So these models are already, methods are also there in statistics. But when coming to machine learning, we have one more added advantage here is that once a model is being generated or trained model is generated, tomorrow when we have new data, You need not to start from scratch. The model itself tries to train themselves. I'll give more examples in my next slides. So moving forward, you can also look at different data that is used in MLS. When you have a design of experiments, you carry out a lot of experiments, and then you try to predict the data from the actual values. That's where we try to generate the models. But this modeling is being on active learning process. So machine learning means we try to learn each time so that to improve and to enrich whatever that we have learned from the past. That is what machine learning is meant. So from the new data that comes in, it goes on active learning to minimize the number of experiments and maximize the materials property so that we come up with a better optimization from the models. That is the ultimate goal of these machine learning models in research. But when you look at the reactions or in-house database, we wanted to understand what are the failure or what are the success reactions. So I don't know whether we are maintaining that, right? Because most of the time we publish only the success reaction. The failed reactions, it is just an experience for us, but very few people report it, where most of them does not report it. I would recommend the first thing that you have to report is the failed reactions only, so that the others don't repeat it. And for you, it's an extra paragraph to explain about. And in machine learning, it's very, very important. Which condition or what type of conditions made the reaction fail, or it failed, or what it made success? That analysis or that modeling really helps you to optimize your future experiments. And similarly, if you're working on some computational chemistry or quantum chemistry calculations, of course, they take a lot of time. And similarly, you can use machine learning in order to calculate from the data from simulations. Now, if you ask me, can we do MD simulations on machine learning? Answer is a straightforward no, because we have a lot of challenges on force fields. The force fields, whatever we are using for a small molecule is absolutely fine, but when you go for a larger system, it's difficult. And when it comes for heavy elements, also it is again a challenge. So machine learning with in ML, like I mean, carrying out MD simulation with ML is difficult, but analysis of MD simulation using ML, that's okay. Now, when you look at the public domain also, let's imagine that you have a neural network to predict the reaction condition. For example, you're giving the reactant and the product, and then based on the earlier analysis and understanding, the model will be able to predict for this possible reaction, these are the following reaction conditions that is suggested by the model. The catalyst could be this, the solvent could be THF, then reagent, and at a temperature 52 degrees. So these are the conditions that we are looking at, which could be predicted by a model, which could also help us to understand. So here, when you're looking at deep learning applications, as I told you earlier, we need tremendous amount of data for deep learning. Don't sit with 40 molecules for deep learning and never do that. Nothing going to work. So we have a lot of other applications looking at accelerated quantum mechanical calculation, QSPR, conformational exploration. And we can also design the molecules by materials design, molecular design, or even drug design. And also we can look at prediction, reaction prediction, optimization, retrosynthesis. So retrosynthesis, it's already being proven how deep learning is really helping us to understand that, right? And also on the QSPR is also being explored a lot more. But the others still it's optimizing, and we have to see a good result. When you look at the QSPR, so when you have the structure and the property, the relevant activity data available, we go for machine learning, try to apply and then predict the data. And then we try to take the error values also, accuracy or ROC or error value, depending upon the type of data that you have. And then we have a training cycle, predict, evaluate, optimize. That's how it goes. And then we come up with a correlation to understand how far the predicted, the validated, and the training set is coming up in par with accuracy. So that's how the models are being generated. But the biggest challenge here for one is the schematic representation of these components, because the output of these analysis comes up with a different schematic representation. Understanding this and interpreting this is something very important. I'm sure you would also have a situation where scatter plot how this to be analyzed, because one set of compounds are in the top, another set of the compounds in the bottom, in between it's completely blank. Should I consider that model or not? So many things can be extracted from those schematic representations. So data analysis is something very, very important. Now, throughout these slides, whatever I told you, you could have come to a conclusion that ultimately you need to know how to analyze statistical data. That's it, because that's where only the machine learning is trying to override some of the challenges that you had during statistics. But the basic understanding about validations and interpretations, it's all same as statistics. This was something that was published where they used some data like TOX 21 data and few others in order to try with certain methods and to see how far the accuracy is there. So you can see some are having very bad accuracy, some having at least more than between 80 to 85%. But when you go for a toxicity studies, we could see around 95% of accuracy is being achieved. So different projects based upon the data availability and based on different models or approaches, you could see the performance of the model will be entirely different. So don't expect that from your data, you will always get a better R square. So it's completely dependent upon what kind of data that you are considering. I have seen many of them discussing with me that, sir, I have this much data. I want to perform this one QSCR. But when we try initial itself, we could see that this data is not going to work. If it is not going to work, don't work. Don't try to make it work and then do it, because that's where we try to remove the compounds as outliers and then do it. So the question is, why did you remove the outlier then? We should have a very firm reason why did you remove the outlier. Ultimately, we know that when we remove the outlier, the model improved. But why? There should be some scientific reason behind it. When you removed, why did the model improve? So make sure that you ask a question like why everywhere when you're doing something in science. Then the discussion and reasonings will come automatically. Now let's see how the chemical structures are being represented in machine learning, or how they are being understood by the computers. I can see that tomorrow you're going to learn about TensorFlow. So where it is going to be much more implemented in GPUs, right? So it is very important that we need to understand how these informations are transferred to GPU or even in CPU. So that's why we talk about fingerprints. So there are several fingerprints available. See, this is all available since 80s. The drug discovery or computer-aided drug discovery started with similarity search. For similarity search, they used daylight or even max fingerprints, because the structures were converted to fingerprints in order to do a similarity search. And we're using the same fingerprints now in machine learning or even in all sophisticated softwares to do certain analysis. So we are still using the old technology. So that's what I was telling you. I have published a review in 2013 for ML in QSAR. So I'm talking about 2013, which is almost 7 years back, right? 7 to 8 years back. So that at time itself, it was there, but it was never hyped like this, what is happening right now. So that time we were mostly using genetic algorithms, support factor machines, or random forest, nothing beyond that we thought of, but in combination with multilinear regression methods and others. So people were back then itself were being using it, but not much hype. So don't forget, ultimately what I want to try to say is that don't forget statistics, because that is the bread and butter for ML. Unless you learn statistics, no go for machine learning. Keep that in mind. So when you come to chemical structures, it is not the fingerprints for a whole molecule as such, but we want to look at the local or the atomic descriptors also. The descriptors, as you know, there are 1D, 2D, 3D descriptors available. based upon the what kind of structures, what kind of dimensions that you're looking at, and what kind of property that you're trying to extract. So here, if you see a keto group with carbon double bond O, this could be the fingerprint for that one. And for this one at different position, this one. And for hydrogen CH, this could be the fingerprint. But overall, the global descriptor for this total molecule could be, this could be the fingerprint. Now that's where we wanted to understand how we can use something without any coding. So yesterday you had used a GUI for machine learning based analysis using Weka. Weka is also there back since 90s and 2000s, widely used in chem informatics before NIME or something coming in. Weka was being very widely used. Good thing is that Weka is also implemented in NIME. Not only Weka, many more other programs. So let us go through that. So I'm also happy to tell you that Nairo Research India is an official partner for NIME in India. And I am also one of their representative who is an expert in the life science and drug discovery domain for NIME Analytics platform. NIME Analytics platform is open access. Anybody can access, anything can be done. It's completely free. But there is something paid version is called NIME server. That is mainly for web deployment. For example, nobody knows how to use NIME. So developers try to generate all the workflows using NIME Analytics platform. And the users can access the models and predict values using the web browser. So that is where NIME servers are. So not only web browser, there are many other platforms. So that is a paid version. But in our case, when we are doing research, we really don't require that. We are pretty much happy with NIME Analytics platform. So this is where we can do automation and we can do data analysis, data integration, and as well as parallelization. The advantage here is that you can always look at the data, and also you can integrate multiple functionalities put together. For example, you wanted to do a 2D to 3D conversion of the molecules. For example, you can use, for that we usually use OpenBabel. Then I wanted to generate the conformations. Again, you can use OpenBabel or Balloon program in order to generate conformations. And then after generating the conformations, I want to prune them. And then I wanted to do a molecular descriptor calculation. And then I want to go for a document. Let's imagine like this. But you might be using 4 or 5 different softwares for this one. In NIME, you can do this all in a single window. That is advantage, to put it in layman terms. So all these packages put together in a single window, and you're just giving input output ports, and then it does the job. Just click a button, all goes automatically. But to make it automatic, ensure that you know what you're doing. Because if you did some mistake, then you're getting a wrong result. So, or it doesn't work. So make sure that it works, it is being well tested, and then you implement it. So most of the time, it has saved a lot of time during my research studies back in 2010, 2011, even 2000. I started using since 2008, actually. So it's very important that the right way you do, actually, you can make use of NIME a lot. So you can download the NIME. Right now, the version is 4.3. This is showing 4.2, but the latest version is 4.3. I have given the instructions in the page where you got the information about the links. Whoever has given the name there, you will be getting the book related to drug discovery, chem informatics, and NIME, which is a very informative piece of information where it can help you to start with NIME. And then you have to, when you're doing the installation, you have to mention the path, and you have to assign the heap size of the memory. So larger the size, if you're working on a larger system, for example, a huge data set, ensure that you give a better memory. It will tell you how much memory it has, and accordingly within that you can give minimum or maximum. Minimum, try to give 1 GB, fair enough. But if you think that you're going to work on larger systems, then 4 GB is also fine. And keep in mind, it all completely depends upon how much memory RAM you have in your machine. And then you have to set your workspace. So workspace is something that all your input output files will output can be saved anywhere, but all your node informations, parameter informations, everything will be saved in the namespace. So workspace. So keep in mind you have enough space in that specific drives, and also write and read access to that folder. Even if any platform, the same rule. Now comes very important part. You have to install extensions. By default, NIME does not have many extensions. They will have only extensions for inputting CSV files or some file reading or file output only. But then you can install extensions. The extensions are of wide variety. When I started in 2010, 9 and all, we didn't have these many extensions. That time I still remember we had only CDK. I don't know whether anyone is still using CDK. Now everyone is using RDKit basically. So that time our favorite one was CDK, and then few other chemistry nodes was there. But looking at bioinformatics also, we had very few nodes available at that time. But now there's a lot of extensions available. We will go through it during the practical session. And then we can create a new workflow. It could be public or it could be private. You can create an account in NIME Hub with the email address that you have given during the download. And then you can save your workflows privately, or you can put it in public so that others will be also making use of it. And you can give a name to create it. Now, when you look at the NIME interface, this is how it looks like. So one of the participants rightly said, it is installed, but looks complicated. Yes, at the initial look, you will feel it is complicated, lot of windows, where to do, what to do, but it's very, very simple once you understand what is the concept here. So let me go through it. So whatever you see here as NIME Explorer, this is what I was telling you, NIME Hub. You can save your workflows in the cloud, in a private or in a public mode. Or even you can download several examples, even related to drug discovery, chem informatics also under examples. And whatever that you have saved, the workflow saved in your workspace will be available in the local workspace. So that is all about servers and workflows. And the second window is called Workflow Coach. So based upon the work, this is called nodes. Whatever that is being put up here square, they are called node, N-O-D, node. So nodes. So as when the nodes are put into the workflow editor, so this is called workflow editor, the white region. That is our main working area. So when you put any nodes into it, automatically it comes up with the workflow coach, which tries to tell you how other people have used this nodes with other nodes. So it's just giving you an idea whether for the beginners, it's always helpful. But for this, you have to share your usage statistics with NIME, because until unless then they cannot pull out the information. This one is called node repository. As and when you install your extensions, you will be able to see the nodes here. So nodes are categorized according to certain criteria. I will explain a bit more when we are getting into the practicals. And then we have an outline. So this is a bigger picture about the workflow. This you can drag and drop, you can drag and view and navigate if your workflow is quite big to fit within this particular workflow editor. In order to bring in the nodes from the node repository to the particular, let me see whether, okay, just a minute. I should show you a laser point. Sorry about that, I forgot. So the nodes that in the node repository, if you want to transform or bring any nodes to the network, or that's a workflow editor, you should click and drag and drop it here, right? Or double click on this, any of the nodes here, it will come automatically here. So these are the two ways. And when you click on a particular node, either on the workflow editor or on the node repository, you will be able to find the description of the workflow as well as of the node. So this is very important because the description tells you whether what should be the input of that particular node and what should be the output, what is the expected output of the node. Very important so that you don't make mistakes. And finally, you have something called a console, which talks about errors, statistics, and other the current state of the node. All those details will come into the console. Okay, now a few other things, like as I told you that you can drag the particular node into the KNIME project or the workflow editor. And when you click on this particular node, you will be able to see the description of the node. And similarly, if you are again dragging, this is what you see, the node description. But when you want to configure that particular node, if you want to make some changes, you double click or configure. That's where you see respective window. And this window can be different for each of the nodes, which we will be seeing in a moment. Now, these are the different extensions that we are going to explore today. So we have extensions like CDK, NLOS, Oldwood, Indigo, LASA Limited, NGS, RDKit, Boanalyze, and all. And we do have some commercial nodes also, which is from Chemaxon, Biosolve IT, Cresset, Molsoft, Schrodinger, and MOE. Of course, unless you have paid licenses from them, you cannot use these nodes. So we are not going to discuss any one of them today. We are going to discuss only about the free nodes today. And these are the different functionalities which is available in many of the nodes. It has docking, it has pharmacophore analysis, it has confirmation generation, 2D to 3D. By the way, are you able to see my screen? Because Zoom was giving me some issues. Okay, I think no issues. Okay.
Yes, sir.
Fine. Great. So, and also CDK, you can look at Lipinski rule of 5, you can calculate descriptors, you can do virtual screening, you can do similarity search, you can generate fingerprints, you can align pharmacophores, many things can be done within this nodes. So these are some of the commercial nodes, which I already mentioned, but in order to work on this, you need to have commercial license, paid license from them. And let me tell you, if you have any unauthorized licenses, they don't work with KNIME. And these are the basic chemistry nodes also. For example, these nodes, chemical sketcher, structure sketcher, and chem sketch, Marvin sketch, you can use it for drawing the structures. And then you have reaction reader also. Then these are all file readers. For example, you can read SDF, mol2, mol file, smiles, all those things can be read by these file readers. And then you have conversion nodes. If you want to convert from mol to CDK or molecule to RDKit format is slightly different from the normal mol or SDF because it wants to go into fingerprints. And then you can have open table. Keep in mind, if you're using Windows or Linux, open table binaries are built into it. You need not install open table separately. But if you're using Mac for open table, you have to separately install open table in your machine and set the path. That is mandatory. Otherwise, open table will not work. And then you have mol converter from Chemaxon, which is again free. And then you have multiple viewers also. You can view the structure in 2D, 3D. You can look at protein ligand interaction viewer, proteins viewer, and pharmacophore viewer. We'll be doing that exercises today. Right, so these are the examples. And these are some of the chemistry calculators. For example, if you want to calculate descriptors. So descriptor calculations are from different providers. Some are overlapping, some are different. Make sure that you join them considering that they are not overlapped. We will be doing this exercise also today. And then in order to write the output file, we can write our output as a table or a CSV or an Excel, or even you can write the pharmacophore information also. I'll be showing this example. We can write it as smiles, mol2, mol, and SDF. So I'll tell you, which I was using earlier during that 2010, 2008 time, how KNIME was more utilized was, if I want to convert 2D to 3D, and if I want to convert smiles to SDF, Rather than using OpenWable, I was extensively using 9 because the pipeline was, the workflow was already being fixed. I need to just give the input file. Automatically, it does all the other job, including ligand sanitization, which is very important. In some of the commercial package, you call them it as ligand preparation. The same thing can be done here. So when you look at the output table, you will be able to see certain identifications, right? So this looks like the header of the column, and this is the row ID. But you can see something I. I means it is integer type. If it is yes, it is string type. So it tells you what data type is there, which is also very important when you try to do some machine learning analysis. So today we will try to do 2D to 3D structure conversion plus geometry optimization using MMF or UFF. This workflow was published in 2018. It's there in my GitHub, the links that was given to you earlier. So you can look at, so I have given you only the picture. You have to do it yourself, right? Because otherwise, how do you learn? But the pictures are fair enough. This picture is enough for you to build a model, so that you have to have a better understanding what you have to do and then try to carry out. And similarly, if you want to predict the environmental and developmental toxicity, which is especially from EPA, Environmental Protection Agency, that is US, right? So from there, you can, what these models are openly available, nothing paid or anything like that. So these are taken from EPA, and you will be able to predict these values. For example, mutagenicity, carcinogenicity, developmental toxicity, skin sensitization, fish acute toxicity, A log P of a set of smiles or SDF or MOL2 files you can, and then you can save it as a CSV file. That is also possible. And then if you want to generate conformers, you can draw the structure, you can load it to convert it to RDKit, and then you can generate the conformations and you can calculate the energies of each of the conformations. Not necessary that it should be one structure. You can load SDF multiple structures, and then you can still do the same process. If you want to also look at the selectivity of the site of metabolism, for example, for P450, we want to see which isoform it's being selective. That can be also explored here, again using RDKit and the LASA's SmartSIP and WitchSIP. These both are published, so you can read more about WitchSIP and SmartSIP. And if you want to do a substructure similarity search, where what you're trying to do here is that I have a database already with me, or if you have two databases is fine, but I have a database already with me. For example, let's say PubChem database. My structure is novel. I'm planning to patent, so I don't want to upload it online. So I downloaded the whole PubChem database here, and I drawn my structure, what I wanted to search. I did generate fingerprints for both of them, and I did a fingerprint similarity. So I wanted to find out whether my compound is similar to any of the compounds in the database. And then it generated a distance matrix calculation, and I was able to view my compound, how far it is similar to, or what is the range of Tanimoto coefficients with respect to other compounds in the database. And similarly, you can do pharmacophore screening. Either you can draw the structure, or you can pull in the zip file, or even the co-crystal ligand in CIF or SDF, whatever file format you have. And then you can generate the pharmacophore. So generating the pharmacophores are important. And then you can align them between, that means pharmacophore screening can be done against the co-crystal ligand or the bioactive conformation ligand pharmacophore along with your database. But this I showed you just a simple example, but you have to generate conformations for this your database compounds before going to screen against the pharmacophore. And if you want to calculate molecular descriptors, that the same thing can be done here. You can calculate it again using different descriptor calculators. And then we will be also exploring orange. Now, what is the main difference between orange and 9? In 9, you can generate the data within 9 itself. For example, for drug discovery related or our properties and other things. But in orange, you cannot generate data. You can only analyze it. And that's the biggest difference. So if you have already generated descriptors or calculated descriptors using PADL or some other packages, for calculating them after or any experimental properties, you can bring them into Orange in order to do machine learning based analysis. So you can download Orange from this website. Just search for Orange software, or the links are already given. You can download it. And this is how Orange looks like. So it's mostly for data analysis. It is not for data generation. But in 9, it is both for data generation as well as data analysis. So you can do feature ranking here. I told you, imputation for imputing the missed values to visualize all the data points. That is possible here. Again, here they use genetic algorithms. Then we have classification trees available with viewer and scatter plots. Then we can do cross validations, including confusion metrics if you are applying random forest classification, logistic regression, and SVM, not for numerical data. And then we have something called Anaconda. So whatever day before yesterday, I think, the session on the protein data analysis using Jupyter Notebook and Python. So the package that you would have installed is Anaconda. Or if you don't have too much space in your laptop, of course, you can install all these packages separately if you know how to configure pip. So I think for tomorrow TensorFlow also you will be using pip and Jupyter Notebook or Spider IDE. So Anaconda also can be used for many of the chem informatics work. And this can be also downloaded. Ensure that you're downloading the individual version of Anaconda. So the latest version is a 3.8 Python version, but I would recommend not to install this for chem informatics and drug discovery work. At least go back to the previous versions and install Python 3.6. Many of the packages are still working on 3.6, and some in 9 still works on 2.7. So today I'll show you how both has to be installed in the same environment. Otherwise, some of the Python integrations within the 9 will not work unless you have 2.7 Python. So you can still install 3.8, but again, you need more space to install different Pythons for 2.7 as well as 3.6. So rather better install 3.6 and use to install 2.7. So we don't require 3.8 as of now, because many of the packages are yet to get upgraded because Python is going fast with so for many years it was 2.7 and suddenly it went into the 3 versions. This is how the navigator looks like, but let me tell you honestly, I never use this. I mostly go with the command line and either Jupyter Lab or Jupyter Notebook. So one of the speaker on the protein data analysis, she was using Jupyter Lab. So Jupyter Lab is fair enough. You can have all things, data can be very well managed within Jupyter Lab. And these are the certain selected libraries that could be used. Keep in mind, these are very important libraries and no need to note it down. All these slides are already available in SlideShare, so you can take it from there. For molecular dynamics analysis, you can visualize the things, NumPy, Matplotlib, Scikit, GROMACS, and even, many others are there. So if you have any doubts on installation and others, there is already a video that is being in my YouTube channel where I'm not a YouTuber, but trying to minimize, because I have plenty of interns and research students, so I started making content for them so that I don't repeat every time how to teach them. So many of the key things are being focused, and you can take the installation help. And if you go to GitHub, there is something called basics. If you go to basics, there will be science. Under science, you have a little bit more information about how to install Amber tools, GROMACS, Python, RDKit, MD analysis, everything within the Anaconda itself. So let me see which portion it is that. Yes. So if you go to basics, you can go to science. And when you go to science, you will be able to see the commands, how to install each of these packages within the Python Anaconda environment. So you will be able to run all these things within the Jupyter Notebook. So why I'm specifying on Jupyter Notebook? Because 9 and Python has a good integration. 9, and Jupyter Notebook also has an integration. So if you have all these packages, they can always talk to each other. So Jupyter Notebook can pull out data from 9, and 9 can pull out or run data what is there in Jupyter Notebook. So that is a reason why I'm trying to mention this here. So these all commands are working, it's all tested, so there shouldn't be issue. Only challenge here is that compatibility of the versions, because this I would have prepared maybe 1 year back. That time everything worked. Now the versions have increased, like for example, Anaconda with Python 3.8. Sometimes that 3.8 might not work because certain packages might require 3.6 or 2.7. Just search something and you will be able to understand that we need to create a new environment for Python 2.7 version or Python 3.6 version. Accordingly, you will be able to understand that. Are there any questions or something? No. Okay. Jyotirmai, if you have Python 3.8, absolutely fine. I'll show you how to install 2.7 in your computer. So it's a one single command step. So don't worry. And the installation instructions for Anaconda, everything is given in the GitHub. Now, finally, before ending my slides, very important, and this is the reason why I have not given the workflows as a file, rather I have given as a picture. Because just copy pasting and taking the workflow, we don't really understand what we have to do. Because having a better understanding about how to manage these nodes, how to face errors and correct them is very important in these kinds of activities, especially when you're working on Python and other integrated machine learning packages. So that's the reason why I have not given you workflows straightforward. Rather, pictures are given, do it yourself. It works. There's no doubt about it. Only thing you have to use a little bit of understanding about what is input, what is output, all those things, right? And also make sure that until you try yourself, see, I'm sure a tremendous of information is being passed. I'm really very much thankful to Vignan University, including Dr. Abram, who is being in touch with me for the past 4 months about this program. So it's a great initiative because, and you will never get this piece of information anywhere else altogether packed. So I know you're getting a lot of information on daily basis. Until unless you try yourself, this will go away from your mind. So make sure that you try yourself, and you also try to teach others, because when you're teaching others, you will also learn yourself and it will register in your mind. That's what I used to tell. As much I repeat the same thing always, I'm getting more expertise on that. It might be boring to a person who would have heard several times, but that's where you become an expert. So until you try yourself and train others, you can never be an expert, because expert can understand what to be troubleshooted. Very important. Nowadays, anything will have some issues, whatever you do. So we need to know how to overcome that issues. That is very important. So that's all from me for as a presentation point of view. And now we will get into practical. But if you have some questions, or if you require a break, I am happy for that. If you have any questions, please go ahead. Or if you expect something specifically that to be showed during the wet lab session, during the practical session, please let me know what are your expectations, what examples that you exactly would like to look at.
Jyotirmai Dev is asking, she's having Python 3.8, sir. So what she has to do, like that she's asking.
Yes, Jyotirmai, I already mentioned that
Yes.
I'll show you how to install 2.7 environment. So don't worry.
Some other is asking about, can you elaborate on molecular fingerprints code?
Yeah, during the practical session, I'll show you some examples.
That's it. What is the protein-protein interaction practical, Bharat?
Okay, Bharat, that is a different topic which won't be covered today. we have to do a separate session for that. Protein-protein interaction studies, many things has to be looked at about the contacts, shape, electrostatistics, and the key amino acids, energy criteria, hydrogen bond criteria, molecular dynamics outcome, many things has to be looked at. So we cannot use NIME or all these things for that. But of course, you can use Anaconda or Jupyter Notebook for that, no doubt about it. But today that was not our agenda. So probably we have to look at it at a different time, unfortunately. Synthetic accessibility is a big problem in silico lead discovery. Is there any node available in NIME to identify synthetic feasibility of a molecule? Yes, unfortunately, they are paid. Synthetic feasibility and accessibility can be analyzed using NIME. Mostly are paid nodes. The free node, one is there, but the accuracy is very bad. So I don't recommend that. If possible, how to create workflow for screening library molecules? Yes, Abarna, I'll be showing you that during the practical session. Yes. Basically, I have certain workflows which helps you to download the Campbell database or even PubChem. It's called RESPIs, and then accordingly how we can load the data into. Yes, it is possible. I'll show you that. But keep in mind when you're using NIME, you require internet if you are working with public database, and you require good amount of RAM if you're working with large data sets. Very important. Only thing that you can avoid here is two things. One, command line executions, scripting, and two, Python scripting. Many of the things are plug and play, and you can just define the configuration. Still, you can integrate Python. I don't say no, but most of the time you can avoid it if you're really not required to do that. So if you want, we can take a 5 minutes break and then start with a practical session. And during the break, I think everybody can concentrate on installing the NIME. And when I'm doing the practical session, I ensure that, please ensure that you're not doing with me. Please don't do that. You will be having all these recorded videos and everything available. So don't do the NIME session along with me. You just watch what I'm trying to do. Please note down the key points or something that I'm clearly mentioning where you have to take care, and then you can later do it. All the data is available in YouTube also. Most of the things that I'm presenting today is also available in my channel. So don't worry about it. So just listen to the session, what I'm going to deliver, and then you can try it later.
Thank you, sir. We'll again meet in 5 minutes.
Yes, thank you.
advantage of the distributed executors on KNIME servers.
Okay, let me know when we can start.
You can start, sir, whenever you would like.
Oh, okay. Okay, so I'll start with the installation part and other things. So you can go to nime.com and you can go to software, and when you scroll down, you will find two things. Don't go to NIME server. We don't require, which I told you already. We can get into NIME analytics platform. You can click on download NIME and fill in all your details. And where did you hear us? Of course, you can mention others, and I told you to mention what to be mentioned, and then you can download it. And when you download it, ensure that when you go down, you can see Windows, Linux, and Mac. You can get the first one itself. But the problem here is all these versions are only 64-bit. If you're still using 32-bit, you have to go click here, like to go to the previous version. Maybe we can say compatibility. I already have my email registered here. So you can access the previous version. So I would recommend you to go to, let me show you, the 32-bit will work pretty old, in fact, but no other choice. If you have only a 32-bit machine, then I think 3.4 or something. Yeah, it's here. I missed it actually. Yeah, from here. So you can install this version 9.4.1.4. You have the 32-bit versions here for Windows. But again, when you're going for Linux, you have to still further go down, and you have to go with again pretty old. So I'm sure you might be not using 32-bit Linux versions anymore, because many of the packages are for 64-bit only now. But Windows you can get from 4.1.4, but for Linux you have to go pretty old. But again, it depends. So let me see if I can still find. I know it's pretty old. Yeah, this one, version 3.3. Yeah, version 3.3, 2018 version. You have versions available for Linux 32-bit also. Otherwise, 32-bit Windows, you can take it from 4.1.4. So don't download the standard one, because that is all 64-bit. So keep that in mind. And the one is that after installations, I told you to start an account in Hub, NIME Hub. So in NIME Hub, you will be able to get many sample workflows, extensions, components. Components are a group of nodes, and individual nodes also can be taken. So you will get a lot of examples also. So if I go to workflow examples, there will be many examples here, right? And I will try to take something related to RDKit. So if I search for RDKit, I can get many, around 151 examples in which I have a lot of workflows available. Right. So from patent, if you want to take the compound, that is also there from RDKit. So certain exercises could be done easily here. And if you click on more results, you'll get structure standardization of molecules, view PDB structures, match to molecular pair examples. All those things can be explored from Hub 9. Even this can be explored same from within NIME itself also can be done. Now, the next part is that Orange, where you can download orangedataMining.com. From there, you can again download Orange. There also, it is mostly 64-bit, but check it out. Old versions are also available, so that you can check that out. If you want to install it using the Python package index, you can use pip install, or from Anaconda, you can install through Anaconda also. So both ways it will work. For Windows, as both options are there, as well as for the Linux. Now, this is the GitHub page of where this is how it looks like, and you can go to repositories, right? And under repository, if you scroll down, you will find NIME. If you go to NIME, only 2 examples I have uploaded. Rest of things anyway, I'll be showing it. So when I go with the 2D to 3D, you have a picture here, PNG file. If you click on that, the picture comes up. So by seeing this picture, manually do it by yourself. Or you can download this picture also, so to keep it as a reference. But I'll be doing this exercise today anyway. And similarly, we can also go to another repository called the QSAR, this one, QSAR. So go to QSAR, and you will see Orange here. In the Orange, there is again one image. Click on that image. You will see the Orange workflow also here. This also you can create by yourself. The only thing that you require is the data file, that you come up with your own data. You can download it from Campbell with activity data, all those things. This is what we are going to do today anyway. Okay. So these all sample files are already available in the GitHub. So let me close one by one. And the slides, whatever I'm discussing today, is also available in SlideShare, where you can go to this particular slides, where whatever today I shared, all the screenshots, whatever I discussed today, it's all available here. And if you want to little more go through with the examples, that is again, these are the slides which I shared today and few more others. And some of the NIME examples I have discussed here under applications of NIME. So don't worry, these slides are all readily available. So whatever today I presented, these are the slides. So they are all available for your references. Okay. And when you're installing Anaconda, I told you I have to go to Anaconda and then under products takes the individual edition. And then 3.8 version is something standard everybody has, where it will be okay. So here you have 2019 and 2020, early 2020 versions. So that will have 3.6 Python. Right. And if you wanted to install something, or if you don't know where to install or how to install, that's where you have Anaconda packages. I should have a search button, maybe under resources. Yes. Okay, let me try that on the kit. Oh yeah, it's under cloud Anaconda. Yeah, right. So now under Anaconda, you will be able to find certain packages and their commands on how to install. So you need to just go to anaconda.org and you will have the search button here, and then you will be able to search. So here it tells you what is a command that to be given to install certain packages. But already all these commands have been compiled under the GitHub. So if you go to Giri Bio Basics, and there you have something called science. If you go to science, you will see the commands on what you wanted to install. At the bottom, you have useful contact packages. So in order to, for the Python integration with RDKit in 9, you have to install RDKit in your Conta pack environment or on your Python environment. Otherwise, 9 will not work with RDKit Python integration. And even modular, GrowMax, Amber Tools, OpenMM, NAMD works only in Linux, not in any other platform. But NAMD as such, it will work for Windows anyway. Other MD triage, all those things, commands are being given here. Okay, so that's all. And this is where you have a few more examples. If you feel like confused, you can check this playlist where you can look at the 9 examples. So this is all about links and other things. So let us, no, Jyotirmay, any platform is fine. NIME is looks same, exactly same for any operating system. So nothing to worry. Okay, so now we came to 9. When you open 9, it will ask for workspace. Just define the workspace. That's all. Then it's done. But then I told you, you have to install many extensions. For that, what you have to do is after opening, you have to go to help menu and then click on install software. After going to install software, you have to click this and say all available sites. So it will search online. Depending upon your internet speed, it might take time. And then it will list many packages like this. Okay. So it depends upon the whatever licensing and other things you have. Don't install everything. It will take too much of space. So for example, if you want something related to database, NBI or extension for bioinformatics, NGS tools, then OpenBase tools or OpenMS, that is again LCMS data management. And then this is protein interface algorithms. Then this is again for SCQA and NGS toolbox. So that is all for bioinformatics if you want to update. Under chem informatics, you have lot many packages like this. Most of them today we will be covering: NLOs, Earlwood, then Genentech 9 features, then HTS, Indigo, CDK, LASA, reaction contributions, RDKit, Wizards, and Vernalis. And then you have other community extensions also. again for deep learning. So tomorrow you're doing TensorFlow. TensorFlow is also available in KNIME anyway. Then you have other community extensions again for AI, MATLAB, R programming, then Python extensions, and many more is available, including some lab extensions available, right? So how to get all this now? This is the question here, right? So for that, a few things has to be done. Before you do this all sites available, go to manage and ensure that you enable this experimental, right? This one will be by default will be unchecked. You enable it, apply and close, and then it will come all available sites. I have already installed in mine, but just for the purpose of demo, I'm just showing it. So it has to come now, or I will redo it. Yeah. So now it is fetching it from internet. What are all the possible updates and anything is available. And then what you have to do is let it come. For the purpose of this demo and other things, what we need require is the complete cheminformatics package. No need to specifically select. You can select all of them. And under bioinformatics, you can select just this one. If you are much interested in NGS and others, you can try, but make sure that you're making use of it because they all take space. So I'm just enabling this one, right? And the complete, oh, sorry, just this one, and the complete cheminformatics package. And mostly I don't require anything else, but still I require one more thing, which I was telling you earlier. We need a Python integration, right? So let us find out the Python one. Let me, if you're finding it difficult, you can search here, Python. So it will, and then press enter. It will search and tell us something that is related to Python. Yeah, so I want this Python extension also to be enabled. And then you can also enable Marvin, which is to draw the structures. You can search for Marvin. So it will come up with the Chemaxon, and that is also free. Yet to load. Yeah, I have a lot of data included, so might take some time. But in the meantime, any questions? No. Okay. So you have to enable this also, okay? Python integrations also. Oh yeah, it has loaded, so I have some issues. So you have to enable this Chemaxon Infocom Marvin extension feature, so that that also will be taken care. And then you have to click next. And when you click next, for me, it's already installed, so it will say it cannot be installed. That's why it is already saying that. Then it will ask you to accept a license agreement. You say accept and next. And once the installation is done, it will ask you to restart. Ensure that you restart, because without restarting, the nodes will be not showing up in your node repository. Okay, so that's all about the installation, and nothing to do much different. The same thing is applicable for any operating system. Now we will start with preparing new workflow. Even though I have examples, I'll show you from scratch how to create a workspace. Okay, so that's how you install and you restart it. So in order to open a new workflow, you can either go to File menu and say New, or in different environments. Otherwise, you can have an icon here, and you click on that icon. It says Select a Wizard. So open a new, start a new KNIME workflow, click next. Wherever is your workspace, in the workspace, this workflow will be saved. So I will say ML demo, right? I can give any name. Ensure don't give any space, either underscore or without underscore. The complete name can be given, and click on finish. Empty workflow workspace will be opened, and there you can generate your things. And now when this is the workspace and this is the workflow in description, you can give the details very much if you're putting it up online or putting it in the public. It is a good practice to include the workspace description. You can click on this pencil and you will be able to add all the details. It is always good to do that. It's a good practice, right? And then now we wanted to, whatever the nodes that we have installed, it will come under community nodes. So whatever nodes we have installed under bioinformatics or even under chem informatics, all comes under community nodes. There are certain NIME lab nodes also. For example, ML interpretability, right? And some other nodes. And ensure that if you're extensively going to work on ML based studies, then you have to also install VECA. So here you have statistics, which is all inbuilt within the NIME. This is the node repository. Okay, don't worry, for you it will at the bottom. So for better visibility, I just made it in the top. And then you can also look at certain manipulations on different row, columns, sorting, merging. All the normal things can be done here together. Now, if you're looking at VECA, we have VECA here, and a few other chemistry nodes are also available for similarity studies. And then we have Chemaxon, again for Marvin, for the Marvin viewer and sketcher. And again, we have PADL descriptors also here. PADL descriptors have issues with the latest version of 9. So make sure that you're using the old version if you're really keen to work on PADL. And this is VECA. So all the VECA algorithms and functions, predictors, they're all available within the NIME itself. So VECA is also very well integrated into NIME. And if you want something on R package, and that is also, I think I have not installed it. No, I have not installed. Yeah. So if you install the R package, yes, if you install the R package, that will also be coming and showing here as a node. So if you have to install it. Again, the installation is still the same. Go to help menu, install extensions. Now let us do the first thing. So I will go under the chemistry, input, output. Yeah. So this is my empty workflow. Either if I want to read a SDF file, right? I can, when I click here itself on the right hand side, you can see the description. Please go through this, spend some time. And here it is said, your output is the table with the molecules that has been read, and output, another output will be which is having errors. So always the readers means you're reading from a file. So if I double click on it, it will come here. Okay, if you want to delete it, just press delete button, or you can just click drag and put it here. These are the two ways you can load it into the workflow editor. And then you can click the right button and say configure, or you can click the right button, or sorry, you can double click it so that the configuration file will open. And here you have to browse and take your files. The SDF files, you can take multiple SDF files, not a problem. And go to the property handling to see what all the data is there. So if you scan, when I scanned both the files, I could see there are 30 molecules available. You can see here, analyzed 30 molecules. If you think that I don't want all the data to be loaded into my SDF file, I can deselect. I will say I need only HERG, log-S, and probably molecular weight. I don't want any other values. And then I give OK. Now there is also a traffic light signal system here. Let me go to right here. Okay. So here you can see there's a red light here. And if you go here, the input here is, it is clearly said a table with SDF or a mol file column. If you have that as an input, then only this will write. Now, what is this writer means? If you double click it, it is saying no SDF value. So that is error. It wants some data. Without that, it will not show you any requirement. So in order to do that data, what we are doing here is we are reading an SDF file as an output. We are giving an SDF file as an output. So I'm going to connect this output file. So I just click on it. And then I take I might drag it and connect it to this particular input. So the output from this file, SDF file, will go into as an input to this SDF writer. Now, if I double click, it got the information. I can browse and I will say I can save this file as something else, maybe parent or something. Or if you want to change the name, you can change the name here, 119 demo. I can save it as SDF file. You can tell what all columns to be considered. So you can see these only the 3 columns I considered for the previous to extract. I didn't ask to extract all the columns. But here, if you still think that you don't want all this column, you want to remove, you can remove them one by one or all of them together. So these are all basic concepts that you have to keep in mind for any nodes. It is something that is same. Another very, very important factor, not for this one. Let me try for this one. In certain nodes, you will find something called as memory policy. You can go to that tab. If you feel that your machine is having very low memory, right, like 2 GB or 4 GB, you can enable this write tables to disk. Only thing, your hard disk space will little bit go up because will be utilized, because whatever that is going to be stored temporarily on the memory, it will be writing it to hard disk. Slightly slow, but it will do the job, so that you don't have any challenges on your, if you have limited memory. So you can write tables to disk can be enabled. For me, it's okay. So I'm to make it faster, I'm doing that. And then I can write this. So here it is red color. Red color means it is in need of data. If it is yellow color, all the data is sufficient. It has to be run. So once I don't click any of the node, you have something here. You can see this double play button. So that is called execute all executable nodes. If I click this, the first one, this node will run, and then next, this node will run. If I want to run one by one, I can click this node, click the right button and say execute, or click the button and click on just single play button, execute. So now it has become green color. Now after becoming green, you can see here also at the end, no destination location provided. Please enter a valid location. So that is telling for this one actually. Let us try, because here you can see the play button is not coming because it is not valid. You can double click again. Yeah, the location I gave, I didn't save it actually. So I'll do it once again. And then I will apply. Okay. So now, oh yeah, sure. Sorry, I made a mistake. So if I'm selecting the existing molecule already, ensure that you click on this overwrite. Okay, then only it will overwrite. Otherwise, it will not overwrite. Otherwise, create a new file. I'll say demo. Click on apply and okay. So now it is yellow color. So all the data is sufficient now. Then I click on this yellow. I mean, that's SDF writer and play this button. So it is now written. So if you want to read that file once again, or if you want to view that file once again, let's bring the reader once again here. And then I double click. I will take that this demo. This is what I written now, Tuesday, 25th May. So I'll take it up and go to property handling, scan files. Yes, because I have taken all the data here. So if I look at the read molecules, sorry, I didn't run. So I have only yellow color. Unless it is green, I cannot look at the output. So I click this and I click the play button, click the right button and say red molecules. So even if you're not connected, if you click on the same node, right button at the bottom here, these two will be mostly output, or here something will be coming as output, which I'll be showing you in the next few workflows. So I can click on read molecules. Now you can see my row, the structure. Interesting. Why it is not showing the structure? Probably it is still reading. Okay. It should have been shown. Anyway. Okay. So then all the other values will be shown here. Okay, depending upon what you have selected. Here, anyway, I have selected all the data to be copied, so that's why it has come. So this is a normal manipulation, how you can try with the data and all those things. Now, when you're coming to Hub, okay, I loaded a lot of workflow, so it crashed. But do you have any questions so far? Yeah, this is how it will ask for workspace anyway. I have to load all the workspaces once again. Yeah. I think I loaded too many workspaces. Okay. Yeah, that's okay. Ignore. Kripanati Sri Rama, shall we predict cytokine storm? I'm very sorry. Could you please educate me about this storm, cytokine storm? Sorry, so that I'll be able to comment it. Okay. So let's start with the first example. So this is what is there in the GitHub, so that it will be able to we'll be able to run the show. So first thing is I have already prepared a mol file reader. So by the way, RDKit you should have installed, then only all these modules will, I mean the nodes will work. Right, first I have to read either a SDF file or a mol file. So you can connect it here, right, like this. So I just kept it for reference. If you use any one of them, you can use. So you can have multiple output, but input can be only one from one source. That is very important. So right. So I can take this input output and give to as an input here. So I can, from one output, I can have multiple going to different inputs. But if I am giving a input to this particular RD molecule, the other one will get replaced. So it will accept only one input, right? That's how it is. Then, so I have read the SDF file. I already have an SDF file that is already been given. I have it here. And then I want to convert my molecule to RDKit. So I will say read is SDF molecule. Then I wanted to create a new column called as RDKit. And I want to remove the source column. I don't want the actual molecule because it's getting converted to RDKit format. And if there is, if you need to generate only 2D, if you are loading a 3D structure, you can generate a 2D structure from here. And then in advanced, you can say whether to keep the hydrogens or whether you want to do a partial sanitization of the ligand, which I will be doing in the next workflow anyway regarding the sanitization. And then we have to add hydrogens. If there is no hydrogens, you can add hydrogens, right? And then you can do optimization. So I have the RDKit molecule here. I can use either universal force field or MMFF94. Which force field you want to use, I can use that force field. It will tell you the energy value as well as the converged to 3D structure. And under the advanced, you can mention how many iterations has to be done for the optimization. So this is not a DFT, but a universal force field based optimization. And then you can interactively view that table, or you can write it as a CSV file or save it as a CSV file, or you can save it as a SDF file, or even you can view it as a 3D file. So let's do that exercise. So after connecting all these one by one, now you know how to connect and all those things, right? And then this is how it is being created. Now, if you think that I wanted to generate using a different, not RDF, not RDKit, but I want to use CDK for my optimization step, then you can use CDK also. So this way, this is also, you will get an understanding what's the difference between the 3D geometries. So here I used 3D coordinates and I said, again, CDK to molecule, and then we will view that also, okay? Unless you define where to save, there will be an exclamation mark. That's okay. Now I just click this because I want all to be run. So I click this. So you can see one by one it is going to run. It's run. It's finished. Now let's see the data here. So after reading the SDF file, you click the right button, you can see the red molecules. Let it load. Yeah, these are the structures that is I have loaded in SDF file. I have added hydrogens. Let us see the output with hydrogens. So these are having hydrogens here. And then we optimized our geometry. Let us go to an interactive table to see how it looks like. So this is the energy value, and this is converged using the universal force fields. And then this is the 3D molecule. Now we want to view it, right? We want to see the 3D. So you can go to this 3D viewer, click the right button and say view 3D view. When you click that, the 3D view will get enabled. Let me just simply maximize it. You select which is the compound, you will be able to see the 3D view, right? So this is a universal force field optimized 3D structures, right? So this is for 2D to 3D conversion. So this is a very simple example, right? So once you install all your things, now you need to know, how do I find this 3D view? Because you didn't share me the workflow. I have only the picture. So how am I going to find out this 3D viewer? Again, simple. Under the node repository, there's a search window. There you can type 3D viewer. You got it. There it is, 3D viewer. So from there you can get it. Or you just simply search viewer. All the viewers will be here. So here you have even Ligand's viewer also, then 3D viewer also. Let's try Ligand viewer. I don't know whether it works, but let's try. So I brought in a Ligand viewer. Okay. And then let me see whether I can view the ligands from here. And yeah, it's all given. Columns with labels row ID and click OK. So I just corrected. So make sure what you're trying to give. Here it is saying column with SDF or mode. So it is a RD kit. Then it is asking which one is having the label. It is the row ID. And then I click OK. Then I'm running it. It is run. Click the right button and say interactive view molecular view. Click on this. It will take some time because it's all based upon Java. I don't see anything. Oh yeah, it's here. So when I click on row 0, that compound should come up. Being it's Java, unfortunately, it will take some time. Still, I'm not able to see or show all. Yeah, I really don't know. But I will show you the pharmacophore because that was working fine for me. So ligand viewer, sometimes it is having some issues. But anyway, we have 3D viewer available. So we will look into that. So similarly, so this is for 2D to 3D. Okay, now getting into the next workflow. The warnings are fine because because idle and configured. So here we are trying to do a ligand preparation, like ligand sanitization. What we did is we took a SDF file, we converted to RDKit. We wanted to remove salts forms. You cannot do docking or QSR on salt forms, right? So small fragments where you have HCl or NaCl in a bigger compound, those can be removed automatically. And then you have to add hydrogens. In order to convert it to 3D, you optimize it, and then you can view it. In other similar simple ways, you can use OpenBabel and then make the conversion. But coming to OpenBabel, I told you already, for Linux as well as your Windows, the executables are included. But for Mac, you have to install it separately and mention the pathway. So this is my path for OpenBabel. And I've selected, this is my molecule that has come, that is from the source is SDF. Now, SDF is my input file format, output file format, whatever is Babel supports, all will be listed here automatically. If your Babel is not installed properly, this output molecular format will not be listed out. And if you want to give some options, I hope you all know about OpenBabel options, -m or gen 3D and minus H. So it tries to convert 2D to 3D, and it also adds hydrogen. So this can be also being given. And then you can view it. Another one is you can remove feature remover. This is again, if you scroll down to the end, you can see this is the NIME integration provided by EPAM systems. So you will be able to see from where this has come. So if you double click on the remover, you will be able to see that whether you want to remove chiralities, histrons, R sites, pseudo atoms, repeating units, minor components, that is called the fragments, isotopes. So those things also can be done for sanitization. Or if you want to ignore the stereochemistry errors, those can be also done. And then standardizer is there, where you can standardize your ligand molecules with certain other isotopes, consider only the largest one, neutralize the deuterions. All those things has been taken care. So remove the largest. So you can define what you wanted to consider. So you click this, click add, so that this will be taken care. So whatever in this particular excluded will not be taken care. So whatever you are adding into the right hand of the sanitization. So this is how it is being done. So let me start running this. So yeah, after generating the 3D structures, or if you already have a 3D structure, if you want to convert it to a 2D structure, you can still have a node called generate 2D coordinates. And if you double click on it, you have to say which is the geometry you want, because I want the 3D geometry, that is optimized geometry, and then I wanted to convert it as a 2D here. And then I wanted to view the Marvin view, which is with the coordinates. So in order to run all this, I'll just click on this execute all, click it. So all will be running. So now open table is running with the minus gen 3D minus H. Let it run. In the meantime, let us see output here. So if I click on right button and say view Marvin, yeah, some Java issues. That happens if your Java is not properly installed, but normally it should be fine. But if you want to see here, click result table, you will still be able to see all the different stages. So this is the original molecule. This is if there is salt form, the salts is removed, addition of hydrogen, and molecule that is being optimized. So all different stages can be seen if you have not removed the sources, and also the energy of the optimized structure. Similarly, you will be able to see from here open table, click the right button and say output table. You will be able to see the coordinates that is being generated. These are the 3D coordinates, right? From 2D to 3D coordinates, it's being generated. Oh, by the way, let me see whether there's hydrogens also. I think I'm not able to see that. Yes, hydrogens are also added here. You can see carbon, then all the hydrogens. So because I gave minus gen 3D minus H in open table, so hydrogens are also added. So there are several alternate ways. It's your choice or it's your preferences. And here also we have standardization. Let's see what are the standardized molecules. So here you have standard, because I didn't ask to add hydrogens here. So only the standardized molecules are available. Whatever I have included the property, that only will be coming, others won't be coming. So these are the different ways you can, for the ligand preparation that could be carried out. When you're going for next, this is for confirmation generation. So let me close this so that I'll get some free memory. Then you have confirmation generation. So confirmation generation, either you can bring in a SDF reader here. So I can say SDF reader. So you SDF reader. So you can connect SDF to this one also. You can do that, or structure sketcher to SDF. So double click on the structure sketcher. You can see you can draw a structure here, right? After drawing the structure, You can, if you want to save, you can save that after. So you have multiple drawing tools here. One is Structure Sketcher, and another is Marvin Sketch, right? And then you will be converting your molecule to RDKit, and then you wanted to add conformations. When you click on Add Conformation on the right-hand side, you can see here, you have to define certain parameters, and then your input file is RDKit, and your output will be the calculated conformations. Let us go through it. When you double click it, this is your input molecule, right? And then this is your reference column, which tells how many conformations to be generated. So for the demo purpose, I'm doing only 10. You can give more, or maximum number of tries to generate the confirmation. Random seed, make it default -1, and RMS for keeping -1.0. And when you go to advanced, you can also look at the chirality, torsions, planar aromatic atoms. All those things can be considered, so that, and also we are using UFF to perform the cleanup. After generating the conformations, we can view the result table so that we can see. Let me show the structure. Yeah. So these are the possible final conformations that is being generated. I can say how many conformations required, right? But I generated finally only 100 of them, one of them, selected with one among them. And I'm trying to calculate all the force field based energies. So I click the right button to see the force fields. So I can see the energy difference between MMF 94, MMF 94S, and UFF. You can see there is a major difference between MMF and UFF. So again, a lot of analysis can be done on conformation generation. This is a very normal, a very simple workflow for generating the conformation. Then you have something called as templated conformation, so that you define based upon specific RMSD and understanding with a template. So you can define which could be the template molecule in order to generate the conformation. For example, when you do this template docking, something similar to that. So you define a co-crystal ligand, which is your template, and then based on that, you can generate. So I don't have it here, but simply I'm just giving a template, and the mol block, I can say I'm not defining anything, and then click OK. But yeah, since I don't have the template conformer being defined, it is giving an exclamation mark, which I have not defined it here. So that way, your template confirmation also can be done. More explanation on that, as well as the